Have you ever stopped to consider what truly makes you? What are the fundamental building blocks that define your individuality, your traits, and even your potential? The answer, surprisingly, lies within a microscopic, elegantly twisted ladder, residing inside almost every single cell in your body: DNA. This remarkable molecule holds the key to understanding life itself. DNA, or deoxyribonucleic acid, a term you've likely heard before, is essentially the ultimate instruction manual, the complete blueprint for building and running a living thing, from the simplest bacteria to the most complex organisms, including us. Think of it as the source code of life. It meticulously contains all the information, the precise specifications that shapes your hair, dictating its color, texture, and even its growth pattern. It determines the color and clarity of your eyes, the strength and rhythm of your heart. In essence, everything, from your physical characteristics to your predispositions, that makes you uniquely, undeniably you. This intricate code, this biological legacy, is carefully passed down from parents to their children, generation after generation, which explains why you might inherit your mom's distinctive nose or your dad's infectious smile. Traits lovingly passed down through the generations. Now imagine this. If you were to carefully stretch out all the DNA contained within just one of your cells, it would extend to an astonishing length of about 2 meters long. That's nearly the height of an average adult. To fit inside the tiny nucleus of a cell, this immense strand is meticulously and tightly coiled into organized structures called chromosomes. In humans, this genetic library is organized into 23 pairs of chromosomes, totaling 46, each chromosome acting like a comprehensive volume in the encyclopedia of you, detailing every aspect of your being. The sequence of just 4 chemical letters, A, T, C, and G, these are the building blocks of the genetic code, and the specific order in which they appear makes your DNA uniquely different and distinguishable from everyone else's on the planet. But DNA doesn't just passively build your body according to a preset plan. It actively runs your daily operations, constantly telling cells precisely how to synthesize proteins. The incredibly versatile and essential tiny machines that tirelessly work to keep you alive and functioning. These proteins play crucial roles, helping to efficiently digest the food you eat, diligently carrying life-sustaining oxygen throughout your body, and bravely fighting off harmful germs and infections that threaten your health. Without the precise and continuous instructions provided by DNA, our bodies simply wouldn't be able to function, grow, or even survive. In essence, DNA is both the detailed blueprint and the sophisticated software that powers the miracle of life. However, even the smallest, seemingly insignificant errors in this complex code can unfortunately lead to a range of health problems or inherited genetic diseases. Therefore, understanding the intricacies of DNA has not only revolutionized our understanding of biology, but also transformed the fields of medicine and science. Now, with advancements in technology, scientists are rapidly learning not just to accurately read and interpret this complex code, but also to write it, to synthesize DNA sequences from scratch, opening up possibilities we once only dreamed of. This groundbreaking ability opens up a vast landscape of new possibilities and raises profound and important ethical questions that we, as a society, must carefully consider and address. The fascinating story of DNA, its secrets and its potential, is truly just beginning to unfold. Imagine writing a book from scratch, not just any book, but a comprehensive guide to life itself. Think about the sheer scale of such an undertaking, the meticulous detail required, and the profound implications it would hold. It's not just reading it, understanding the narrative, or appreciating the author's intent. It's about crafting the entire story, word by word, from the very beginning. That's the core idea behind synthetic biology, a field pushing the boundaries of what's possible in genetic engineering. It's about designing and building biological systems for a variety of purposes, from creating new medicines to developing sustainable energy sources. And it's the driving force behind the Human Genome Project Right, or HGP Right, an ambitious initiative with the potential to revolutionize our understanding of human biology. Scientists are embarking on the incredible task of building human DNA in the lab, piece by piece, a feat that was once considered the realm of science fiction. They're using chemical building blocks, the fundamental components of DNA, to construct these complex genetic sequences. The process involves assembling genetic code, letter by letter, adenine, guanine, cytosine, and thymine, the A's, G's, C's, and T's that make up the blueprint of life. The original Human Genome Project, a monumental achievement in itself, mapped our DNA, providing us with a complete sequence of the human genome. HGP Wright takes this a step further, aiming not just to read the code, but to create it, to synthesize entire human genomes from scratch. The ultimate goal isn't to make a whole human being. That's not the intention or the capability at this stage. Instead, the focus is on creating functional human cells with synthetic DNA for research purposes, providing scientists with powerful new tools to study disease and develop new therapies. By building DNA from the ground up, researchers gain unprecedented control over the genetic material, allowing them to manipulate and study genes in ways that were previously impossible. This approach allows scientists to truly understand how genes work together, how they interact to create complex biological functions, and how disruptions in these interactions can lead to disease. They use sophisticated machines, DNA synthesizers, to create short DNA pieces, oligonucleotides, with precisely defined sequences. Then, with meticulous precision, they stitch these short fragments into longer strands, gradually building up the complete gene or even an entire chromosome. It's like assembling a novel sentence by sentence, carefully choosing each word and phrase to convey the intended meaning. But in this case, the meaning is a biological function. The challenge is not just making DNA, creating the physical molecule. That's only part of the equation. The real hurdle is ensuring it works correctly inside living cells, that the synthetic DNA is properly expressed and performs its intended function within the complex cellular environment. This ambitious project could ultimately reveal the deepest secrets of our biology, unlocking the mysteries of gene regulation, cellular development, and the very nature of life itself. And in doing so, it has the potential to transform medicine, leading to new diagnostic tools, personalized therapies, and even cures for diseases that currently plague humanity. But it's a complex and careful process, requiring meticulous attention to detail and a deep understanding of the intricate workings of the cell. Each step, each successful synthesis, each functional gene brings us closer to understanding and potentially rewriting the code of life, opening up a new era of biological possibilities. The future of genetics is being built, one letter at a time, in laboratories around the world, as scientists push the boundaries of what's possible and strive to unlock the secrets of the human genome. HGP Wright has both immediate and ambitious goals. First, scientists want to make DNA synthesis faster and cheaper, so more researchers can join the effort. They're starting with simpler organisms like yeast to perfect their techniques. Next, they aim to build complete human chromosomes for research, like designing virus-resistant or cancer-proof cells. The long-term vision grow organs for transplant, perfectly matched to patients, ending donor shortages. Ultimately, building a human genome from scratch could unlock the function of every gene and lead to personalized medicine. This project could help us prevent diseases before they start. The goal is to master our biology and improve health for everyone. The journey is just beginning, but the possibilities are extraordinary. Synthetic human DNA. It's a concept that once belonged solely to the realm of science fiction, but now it's rapidly becoming a tangible reality. This groundbreaking advancement could revolutionize medicine as we know it. Imagine a future where diseases are not just managed, but eradicated at their very source. Scientists are on the cusp of being able to correct faulty genes with unprecedented precision. This opens up possibilities that were previously unimaginable. These faulty genes are often the root cause of devastating diseases like cystic fibrosis, a condition that severely impacts the lungs and digestive system, or sickle cell anemia, a painful and debilitating blood disorder that affects millions worldwide. Synthetic DNA offers the potential for permanent cures, not just temporary treatments that manage symptoms. This is a paradigm shift in how we approach healthcare. Engineered cells could be programmed to specifically target and destroy cancer cells, leaving healthy tissue unharmed. Furthermore, they could repair damaged hearts, restoring function and improving the quality of life for those suffering from heart disease. Imagine the possibilities of being able to regenerate nerves, potentially reversing paralysis and restoring lost sensation. Synthetic DNA lets us program cells for specific medical tasks, essentially turning them into miniature drug factories or repair units within the body. This opens a completely new toolbox for doctors, providing them with innovative tools to combat disease and improve patient outcomes. It could also potentially slow aging, a concept that has fascinated humanity for centuries. This could be achieved by making our cells more resilient to damage and significantly better at repairing themselves, extending our lifespan and health span. The technology could dramatically speed up vaccine development, allowing us to respond more quickly and effectively to emerging infectious diseases. This enhanced speed and efficiency in vaccine creation would make us significantly better prepared for future pandemics, potentially saving countless lives. We might even be able to design cells that are inherently immune to viruses, providing a powerful new defense against viral infections. Furthermore, synthetic DNA could lead to creating safer and more efficient ways to produce medicines, reducing the risk of contamination and improving drug quality. The ultimate promise is longer, healthier lives for everyone, free from the burden of disease and disability. It also offers the potential for rapid responses to new and emerging health threats, allowing us to adapt and overcome challenges more effectively. Medicine is undeniably on the brink of a new era, an era defined by the power of synthetic DNA and its transformative potential. With great power comes serious responsibility. Synthetic DNA could be misused to create dangerous pathogens, raising security concerns and the need for strict oversight. The idea of designer babies sparks debate about inequality and the ethics of customizing humans. Releasing synthetic organisms could have unpredictable effects on nature. Built-in safety measures are being developed, but risks remain. As we rewrite life's code, we must ask, who decides how this power is used, and for whose benefit? The scientific community is divided on synthetic genomics. Advocates like George Church argue we have a moral duty to pursue this research to fight disease and improve health. They believe careful planning and oversight can manage the risks. Critics like Drew Endy urge caution, warning of unforeseen consequences and calling for global public debate before moving forward. These voices remind us the technology's impact depends on how we use it. The debate isn't just about science, it's about our values and the future we want. With such power, responsibility and transparency are essential. The conversation must include everyone, not just scientists. The UK is a leader in genomics and synthetic biology. British scientists are improving DNA synthesis and delivery, making it safer and more reliable. Projects like the 100,000 Genomes Project are helping researchers understand and model diseases in the lab. The UK's strong ethical oversight, led by the Nuffield Council on Bioethics, ensures responsible research. There's a special focus on using synthetic genomics to fight cancer and rare diseases. British innovation is shaping the international HGP right effort. The UK's balance of progress and ethics makes it a key player in this global story. The future of synthetic DNA is being shaped in British labs. What's next? Scientists are perfecting their techniques with simpler organisms like yeast before tackling the human genome. The next challenge is scaling up, automating DNA synthesis to make it faster and more affordable. Alongside technical progress, global conversations about ethics and safety are underway. Pilot projects will focus on specific chromosomes, like those linked to Down syndrome or radiation resistance. Each step is carefully planned with public input and oversight. The journey is incremental, but each milestone brings us closer to rewriting the human genome. Science and society are moving forward together. The next chapter in our biological story is unfolding now. We stand at a turning point in history. The power to write our genetic code could cure diseases, regenerate organs, and extend healthy lives. But this promise comes with real risks: biological weapons, inequality, and unintended consequences. Science must be balanced with strong ethics, oversight, and public involvement. The future of synthetic DNA isn't just in scientists' hands, it's in all of ours. The choices we make now will shape the world for generations. By combining innovation with responsibility, we can harness this power for good. The conversation belongs to everyone. Our challenge: build a future that's not just more advanced, but more just and wise.